and welcome. In this video I will explain to you principal component analysis, commonly known as PCA, and show how we can interpret either 2D or 3D PCA using the well-known dataset iris dataset. So PCA is a powerful test or analysis multivariate technique that allows us to simplify complex data. For example, for instance, our data contains more than about 150 rows or 150 observations. So we can also use PCA to reduce dimensionality of data and visualize patterns that are otherwise difficult to detect directly when working with many variables at the same time. Throughout this video, I will explain why we might need to use PCA, how to select number of dimension. For example, in this case, we have 3 dimension principal component 1, 2, and 3, and how to interpret using a case rule to select appropriate dimensions, how to interpret coordinates, the cosine squared or square of the cosine, and the contributions of variable. Finally, I will show how these concepts are visualized in either 3D like this way or 2D like this figure 2D. Only we keep only 2 dimensions. So let's get started. First, we might need to upload these or load in this packages: Factor Minor, Factor Extra, Plotly for visualizing 3D PCA, and Factor Minor, Factor Extra is very PCA requires these 2 packages. So using the famous iris dataset, PCA it's a powerful technique, and why we usually select this technique. First of all, PCA allows us to reduce dimensionality and to visualize to explain the correlated variables. So in this dataset iris, we have 4 quantitative or 4 continuous variables: sepal length, sepal width, petal length, and petal width. So the first thing we need to keep in mind that PCA is used to only on continuous variables, and it is recommended like this argument show scal.unit to standardized variables because in this case sepal length and sepal width, before we standardize these variables, we need to check whether these variables are correlated between them or not, using, for example, ggpairs function from ggalley. We need first to load ggalley package and then use ggpairs and select iris only between brackets the first 4 variables, because the second one refers to a grouping variable. Using this function, we can draw a correlation matrix, especially Pearson. We can conclude that many of values are significant, so we have significant or correlated variables, meaning that they often convey overlapping information. We can add here, for example, aesthetic graphical, and select color equal to our grouping variable, which is species to color. Now I need to remove this argument and select whole iris, because species is the 5th variable in the dataset iris. So here we can show the all coefficient correlation percent for each group. Many of significant correlation between variables according to each group. So in this case, PCA transforms these original, for example, iris, these original variables into new uncorrelated variables. We have a correlated status between variables. So PCA helps us to transform these variables, these four variables, according to our iris dataset, into new uncorrelated variables called principal components, which summarize the maximum amount or variance or information in the data using fewer dimensions. In simple terms, PCA helps us to understand how we can describe most information of, for example, iris here, to describe, to explain, and to show the most information of this dataset using fewer axes. So now we might need to move to the second step, which is performing PCA using PCA function from factor minor iris and select only the four variables, because the fifth one refers to a grouping variable or categorical variable, and PCA is according or only used when we have continuous variables. And why this argument is added, because we need to standardize it. It's very important. We here scal.unit, it ensures that these variables that all variables are standardized, meaning they contribute equally, for example, to regardless of their original units of scales. This is essential. Why? We need to scal or use, for example, either scal function to transform the data, because this is essential when variables have different measurements ranges or units, as the case here, or the case of physicochemical parameters. Moving now and click to run to show the PCA. This figure shows refers according to variables, and these two individuals, we have overlapping informations here. So we can conclude that these variables are correlated. So before we interpret the data, we need to get the eigenvalue of our PCA. This argument we have named as PCA. So using get underscore eigenvalue between parentheses PCA, we can show or examine the eigenvalues which represent the amount or the information or variance explained by each principal component. Each eigenvalue, for example, for dimension 1, with eigenvalue equal to 2, for example, 0.9, tells us how much the dimension or how much information a component contains. So the first dimension have the high information, and the case rule helps us to decide which dimension and how many dimensions we need to retain or to keep. So case rule states that only principal components or dimensions with values greater than 1, like dimension 1, should be retained. So in our case, we might need to retain only the first dimension with eigenvalue greater than 1 equals to 2.91, yes. Why? According to the case rule, only dimensions or principal components with eigenvalues greater than 1 should be retained, because when variables are standardized. Each variable contributes a variance of 1. So a component have with an eigenvalue greater than 1 explains more information than a single variable. And if our components like dimension 2, 3, or 4 with eigenvalues below than 1 or below the threshold, in this case the 3 dimensions here, explain less variance and contribute relatively little information or little additional information. So in our case, dimension 1 explains 73% of information or of variance of our dataset iris. Here, the first dimension explains 73%, and the second dimension represent or explains about 22.9%. So together, the first 2 dimensions here explain more than 95% of the total variance or the total information of our dataset or our results data frame, which strongly justify using only a 2D instead to use 3D PCA representation. This is clearly shown in this figure. So moving now to understanding coordinate, contribution, and cosine squared. Coordinate, for example, we can name this argument as var, get underscore PCA underscore var, and select var, not individuals, for variable, and use the common argument head to show the coordinate of each variable according to each dimension. The coordinate indicate how strongly each variable is associated with its principal components. In this case, we take, for instance, petal length and petal width and sepal length have a large positive coordinate, means that the variable strongly contribute to that dimension. So these 3 variables, sepal length, petal length, and petal width, have high positive coordinate, indicate that these 3 variables are strongly contribute to the construction of first dimension. Either we have either a large positive or a large negative coordinate, means also that this variable, for example, we don't have a large negative value here. So either we have a large positive value or a large negative coordinate or positive coordinate, means that the appropriate variable is strongly contributes to that dimension. Also, the variables pointing in the same direction are positively correlated. So we have a strong correlation between these 3 variables because these variables pointing in the same direction. When our variables pointing on the same direction, this is refers that these variables are correlated and are highly and positively correlated. Variables pointing, for example, in the opposite, for example, in the negative coordinate here of the first component, are negatively correlated with, for example, the variable pointing in the opposite correlation or positive coordinates. Variables, for example, at right angles, for example, here, sepal width, and for example, taking another example, if we have another variables here, we can conclude that, for example, this variable are weakly correlated with sepal width. Weakly, not strong positive or negative, but weakly correlated with sepal width, either here or here, not here, because here is very opposite, which refers to that is strongly negative correlated with sepal width. In our case, petal length and petal width have high coordinate equal to 0.99 and 0.96. So in this case, first dimension it mainly represents petal size. So moving on to the cosine squared, also known as square of the cosine. Cosine squared here tells us, this values of cosine squared measures how each variables, or how well a variable is represented on a given dimension. When we have a high cosine squared, like for example petal length and petal width, and a little bit petal length, sepal length, we can conclude that this variable, for example, is well represented by that axis, by the first dimension. And the low cosine squared, like sepal width, means the variable should not be interpreted strongly on that dimension. I forgot to interpret or explain the contribution value. For contribution, now we'll let us examine the contributions for each variables according to each dimensions. The contribution of each variable tells us how much each variable contributes to the construction of a dimension. The sum of these values equals to 100%. So petal length contribute to the construction of the first dimension with 33%, followed by petal width by 31%, and then 27% of the contribution of construction of the dimension 1 from sepal length. So contributions sum to 10% for each dimension, like also this dimension is constructed by the contribution of 85% of sepal width here, the first dimension. According to Kaiser rule, we need only to keep the first dimension, because first dimension with or have eigenvalue greater than 1. And if petal length, I think here, and petal width contribute most, in this case, to the construction of dimension 1, can be interpreted as a petal size gradient, the dimension 1, because these two variables contribute to near 65, I think, percent of the all information or all contribution of this dimension. Moving now to draw the PCA, we can draw the results of cosine 2 here, cosine squared, or of visualization the value contribution for each variables using this comment. F-vis underscore PCA and individuals to draw only the individuals, or plot for only individuals, and this for select only to show only variables. Biplot to include both variables and individuals. Color the individuals using species. Select species from iris dataset and add ellipses for each group. So in this 2D PCA 2D, we have points here, either in red, green, blue, refers or represent individuals. In our case, flowers. In any another case, we can refers, or points can be refers to sites, to samples, to observations. And arrows here, arrows, we have 4 arrows, refers or represent variables. Colors, we have 3 colors, blue, green, and red, represents or points to species, either Setosa, versicolor, or virginica. Ellipses show group dispersion. I think we have here overlapping information. Individuals, when 2 individuals are close together, or when individuals close together, like for example, individuals, for example, 17 and 6, we can conclude that these individuals are similar. Also, taking another example, individuals number 108 and 131 close together, so we can conclude these individuals, or these, for example, individuals are similar. Species separation, species along first dimension, species separation along first dimension shows that petal width and petal length discriminant species very well. So Setosa contains or contain only the low value of petal width and petal length, so we can conclude that petal size discriminant the species. Setosa here is clearly separated, while versicolor and virginica here in blue and red, green partially overlap. This confirms the biological relevance of the PCA. But in this case, why we might need to use 3D PCA, PCA 3D? So I think when we have, or when case rule indicates that we might need to keep 3 dimensions. So if in other cases or another case, our principal components have the 3, for example, contains or with eigenvalue greater than 1, we can instead to use or to draw a PCA 2D between first component and second, and other figure PCA 2D between first component and third component. It's recommended to select or to choose switch the PCA 3D to show the 3 dimensions retained after checking or according to case rule. So while 2D plots are usually sufficient, 3D PCA useful when clusters also overlap in 2 dimensions, especially for versicolor and Setosa. I will remove, for example, Setosa and select only versicolor and virginica. I can click here to show versicolor of the distribution of Setosa, I mean not versicolor. So it provides a more complete view of the multivariate structure, include the 3rd principal components here, capture additional variance, and better visualize complex group separation. What we could conclude in this case, PCA is powerful, yeah. PCA is a powerful tool that reduces the dimensionality of our data, reveals hidden patterns, and allows meaningful and huge interpretation of complex data. By applying, it's recommended to apply in case rule and clarify interpreting coordinates, cosine squared, and contributions. We can confidently explain all information of both either 2D or 3D. It's a very useful and high published quality of this figure. I think this is all of this today's topic. If you have any questions or suggestions, please let me know. Feel free to, don't hesitate to let me know below in the comments. All these arguments or the code, I will leave it in the comment below the video. Thank you for your watching, and I hope this was helpful for you. I hope this explanation helped you better understanding PCA and its interpretation. See you soon. Take care for yourself.